Hi, welcome to biology classes with Lakshmi Chandra. At the outset, I would like to thank all my viewers, all my students who are here for giving me such an overwhelming responses and energy to put forward more and more videos. One of you have requested me to make a video on translation as you are preparing for competitive exams. So I had given you a word that next week probably I'll come with that, and as such I'm here with that. So this translation is actually a long process that is happening, but it is very easy for you to learn and understand. Since it is a very long process, in the next week there will be a series of videos on translation. And this translation I have divided broadly into two heads: translation in prokaryotes and in eukaryotes. In both these cases, I'll be giving you an overview first, and then we'll go on to each and every aspect in detail. That will be more easy for you to understand, recapitulate and reproduce for your exams, and also gain good marks and come out very successfully in your future. Let us go back to our session for today. Today we'll deal with an overview of translation in prokaryotes. Translation in prokaryotes. So before going directly into that, we have to see the stages regarding as what it is ultimately leading to translation from the beginning till the end. So that is already dealt in the previous sessions, but still, in order to give you a clear understanding, I feel like I should go back to that a little bit. So in a nutshell, let me just take you back to this central dogma of molecular biology. As a biology student, all of you must be knowing about this central dogma, but still, let me just tell you quickly what that is. DNA is forming another DNA. A DNA template is there, it is producing another DNA based on the information there in the template. So that process is DNA replication. Okay, so that is happening inside the nucleus. Now, from the DNA, the information is transferred to an RNA. Most cases it is mRNA which is formed, and that process is called as transcription. That was also dealt with in detail in my previous videos. Transcription in eukaryotes, prokaryotes, the different stages. So those also you can just go through to understand this particular process. Topic for today is how the information in mRNA is converted to small polypeptides, or it leads to the formation of proteins. So that is translation. Information in mRNA is converted for the production of proteins. It is not mRNA is converted to proteins. mRNA remains there as mRNA only. It only gives information in the form of codons. And using that information, the cell can produce new and new proteins. Okay, isn't that wonderful? That is happening inside our cell. Tiny such things. So our topic for today is this: translation in prokaryotes. Now this whole process of translation in prokaryotes, I'll just connect it to films, okay, in cinema and all. In cinema, what is happening is, in cinema when it is released, we look for who is the hero, who is the heroine, villain, likewise, isn't it? And then director, other cast members and all. Same way, I'm just trying to connect my topic for today with cinema. Okay? And after viewing this, please share your responses so that whether it is good for you or whether it has helped you out or whether I should not do this. Okay, I want your responses. Fine, let me try connecting it. So translation in prokaryotes. Here, the heroine is mRNA and the hero is tRNA. Okay? And the meeting place where they're doing their dream come true. That is, their aim is to synthesize proteins. This hero and heroine together, their job or their aim is to synthesize proteins. And that they are doing at a place called as ribosomes. So heroine and hero is there, and they have found out a place to do the protein synthesis. That is their job. Okay. Now, to help them out, they cannot do it on their own. To help them out, there are many other cast members also, and they are enzymes are there. Here I have named two enzymes, amino-acyl tRNA synthetase and peptidyl transferase. Two enzymes to help them. And there are other factors. Other factors here means other proteins. Okay, other proteins to help them. That is, so this topic of today, that is translation, I'm just connecting it to certain members of cinema. Okay, so that it will be easy for you to understand. So in cinema, as you know, there will be hero and heroine, and there will be other cast members. Same way here, I'm just connecting. Let my mRNA here be the heroine, okay, and tRNA be the hero. Okay, they both got married, and now they have started a job. They have started a factory. Their factory is protein synthesizing factory. Okay, they have to synthesize protein, and they have named their factory as ribosome. They have named their factory as ribosome. And to help in this factory, there are other members as well. There are some enzymes. There are some initiation factors, elongation factors, and release factors. These are all yet different proteins. I'll just first introduce these cast members to you in this video. Okay, the components I'll introduce to you. So once these components are all set, the whole process or the whole cinema, as I've told you, you can just put it under 4 different parts. The first part, the cinema shows about the pre-initiation or the charging of the hero, that is tRNA. Okay, then we can see initiation, that is how this hero is meeting the heroine. That is we are seeing in the next one. Okay, next they got married, and then they have started the factory, and how they are taking their life on, that is elongation. And then we'll see the climax of this film, that is termination. Okay, so the whole thing you have to just keep it in mind like this. So dear viewers, if you just keep it in mind like this, you will never forget things, and in whatever way in the exams things are asked, you will be able to reproduce also very clearly for that. So with that aim, let me just introduce each key players here. Okay, first one, mRNA. You all are familiar with this mRNA, and in this process of translation, always the process occurs in the 5' to 3' direction of the mRNA. Okay, always in the 5' to 3' direction. And what is there in mRNA is actually the codons. In mRNA you have codons. So codons are actually triplets of nitrogenous bases. Triplets of nitrogenous bases. You know, in mRNA, the nitrogenous bases are adenine, uracil, guanine, and cytosine. So these 4 bases will be arranged on the mRNA like this. For example, if I am putting like A, C, C, G, A, U, U, A, C, G, A, like this. Okay, I have put all these bases, these 4 bases in this manner here on the mRNA. Now what are the codons? As mentioned here, codon is a triplet of bases. So these 3, A, C, C, this is one codon, 3 bases together. Next, G, A, U, this is another codon. UAC is the 3rd codon. GAA is the 4th codon. Likewise, it keeps on going. So this mRNA is actually providing the codon. So codon is what? It is triplet of nitrogenous bases. And what it is giving actually, this codon is giving the information to produce the protein. Codon is actually giving the information to produce the protein. And you know, protein is made up of amino acids. So actually this codon is giving what? The information like which amino acid should come first, which should come next, third, fourth, likewise. So here if I have the codon ACC, this is giving the information, okay, from the cell amino acid 1 has to come and sit here first. Then GAU is saying 10th amino acid should come here. UAC is saying second amino acid should come. GA is saying, say, 15th amino acid should come. So likewise, how many amino acids we have? We have 20 naturally occurring amino acids in our cell. Okay, so which amino acid should come first, next, third, fourth, fifth? Likewise, who is actually giving the information? This mRNA, the codons present in mRNA is giving the information. Okay, so remember the whole process of translation. If I say in the case of prokaryotes, there is nothing like nucleus, so there is only cytoplasm, or you say there is protoplasm. So DNA is here, DNA is producing the mRNA, okay, and as the mRNA is produced, protein is also getting synthesized. That is, amino acids are attached and a protein is getting synthesized. So transcription, that is production of mRNA, and translation, that is production of proteins, they both occur simultaneously, and you say both the reaction, they are coupled. The reaction is coupled. Transcription and translation is coupled in prokaryotes because there is no nuclear membrane that is surrounding this mRNA. But in the case of eukaryotes, it is not like that. They have a nuclear membrane and the mRNA is produced inside the nuclear membrane, that is inside the nucleus. After it comes out only, it can do the process of translation. So there it is not coupled. But in this topic of today, that is in prokaryotes, the translation is coupled with transcription. That is, both of them take place simultaneously. One after the other it happens. Okay. So here, see, codons, they are giving information like what should come first, next, third, likewise, which amino acid. And then all these amino acids are joined and then a protein is formed. Now, next comes our hero, tRNA. So I told they both have started a factory of doing. So the heroine is having the information which amino acid should come first, next, likewise. Actually, who is bringing these amino acids? It is the hero, the tRNA. It goes to the area, that is to the protoplasm where these amino acids are there, and as told by the heroine, that is as given in the codon here, the hero brings the amino acids. That is, the tRNA brings the amino acids. So we should have a glance like how tRNA looks like. That is very essential. Let's move on to that. tRNA. That is also an RNA. So that will also have a 5' end and a 3' end. Definitely it should have. But the structure is like a clover leaf structure it is. Clover leaf structure. I hope the diagram is clear to you. This is how tRNA looks like. mRNA is looking like a thread, 5', 3', finished. But tRNA has a clover leaf like structure. If you play cards, you know, clover leaf is the structure. It looks like that, clover leaf structure. So this also I told it is having a 5' and a 3'. We should clearly understand 5' is this, a short arm here. This is 5' and 3' is a bit higher here. This is the 3' end. And always for a tRNA at the 3' end, it will have a region, a base like this, CCA base. And this A will have an OH group here. This always remember, because this is the area where things come and attach. So this is how it looks like. And see, these are the arms with loops, clover leaf, okay. So this arm is called as T-Psi-C arm. This is an unusual base present in tRNA. Usually bases are AG, U and C, but this is an unusual base present. And then here you have the D arm. Here also it has unusual bases here. Okay, this is a loop-like structure. So this is a folded loop, or you can say this is an unusual arm or a folded arm. This is a loop-like structure. Or here the bases can vary with different tRNAs. So instead of unusual loop or arm and all, we can give a refined name. This is also called by these names. Okay, we can call as a different name. You can call this as a variable arm. In most of the books you will find this word, variable arm. And here, this is the area of our importance. This region, this arm is called as the anticodon arm. So we have seen codon, it is present in mRNA, okay, and this anticodon is present in tRNA. So actually this tRNA goes and finds its actual mRNA with the help of this anticodon, that is with the codon. So here, if suppose I have drawn, like earlier I have drawn a mRNA, okay, it is having a 5' end and a 3' end, and it is having many base pairs here. Suppose here the base pair on the mRNA is CCA. If this is the base pair, if this is the codon in the mRNA, not base pair, codon in the mRNA, then the anticodon will have bases complementary to this. Okay, so complementary to C is G, again complementary to C is G, complementary to A is U. So the base in the anticodon is GGU. So this is my anticodon and this is my codon. Okay, so this is how the hero is here, and it is combining with the heroine with the help of anticodon and codon. Next, here it is having another triplet here, that is a codon. Accordingly, another tRNA with the anticodon will be placed here. Again, another tRNA with another anticodon will be placed here. Likewise. Okay, so it is a codon-anticodon is the area where the interaction takes place, according to which they come and sit. Now, I told you here there is a very important thing. This area is called as the amino acid acceptor arm. Amino acid acceptor arm. Or this is the area where the tRNA holds the amino acid and brings it towards the mRNA. I told tRNA is carrying the amino acid as told by the mRNA, isn't it? So here, see, here the codon is CCA. Suppose this codon is asking the tRNA to bring amino acid 1. I am not telling you the names of those amino acids, exactly what is the CCA giving and all, because this was already very clearly dealt with in the genetic codon session. I have taken a full session about genetic codon. What is that and how things are happening. So if you go into that, you will easily understand how to find out the name for CCA, which is the amino acid, AUG, which is the amino acid, likewise. Okay, so here imagine CCA is asking the tRNA to bring amino acid 1. So tRNA, where it will carry that amino acid? On the head part. So here it will carry the amino acid in this region. Amino acid 1 it will carry here, and it will bring and sit over this particular codon, and it will form a bond for the factory to function properly. Okay, and then if another tRNA is coming, suppose here it is CAA, then what will be the anticodon? Tell me. C is here, so it will be G, yes, U, U. Very good. It will be like that, isn't it? So here CAA is asking this particular tRNA to bring amino acid 10. So it will carry amino acid 10 from the cytoplasm and it will come over there. It is carrying everything and it is coming. Where it is carrying all these things and going? it is going to its factory. That is the site of protein synthesis. It is a place where protein synthesis takes place in every cell, whether it is prokaryote or eukaryote. And ribosome is the site where protein synthesis takes place. Ribosome. Okay. So let us now understand about that factory, that is ribosome. See, this ribosome is actually made up of a very small subunit like this, and it is often connected to a large subunit. This is how they are. They are connected and kept. So this whole together is a ribosome. In the case of prokaryotes, in the case of prokaryotic ribosome, the large subunit is having a density quotient, and that is 50s. Small subunit is having a density quotient of 30s. But together the prokaryotic ribosome is having a density quotient of 70s. So it is not the added effect, it is actually the density, and it was found out by a scientist called as Swedberg. So here S stands for Swedberg's unit, or coefficient of its density in centrifugation and all. Okay. So this is the factory where the whole mechanism takes place. So it is having two subunits, 50s and 30s. Okay. At the time of protein synthesis, these two subunits, they separate apart. They separate. So first we look at the 30s, small subunit. 30s. Okay. And the small subunit is having three parts called as the E site, the P site and the A site. Three sites, that means three areas. Okay. Here A site refers to the acceptor site. P site refers to the peptidyl site. So A site is the area where the entry happens. P site is the area where actually the bonding between amino acids, this is a peptide bond between amino acids takes place. That is what is called as peptidyl site. And E site is the exit site, the area for exit. So this is how the factory is designed. A site, P site and E site. So this is there in the small subunit, that is E, P and A. And the same thing is there in the large subunit also, E, P and A. Okay, so at the time of protein synthesis, what happens? They both separate apart. And then now tRNA has to come, mRNA has to come, isn't it? So where in this area of the small subunit of ribosome, mRNA is coming and sitting? Where in this area the tRNA is bringing the amino acids and coming? How they are getting bonded and how eventually the exit happens. So all these things we learn in the next session. So today in this session I have just introduced the basic concepts. Okay, so wait for the next session. Thank you till then.